Assalamualaikum to Madam Munira. My name is SALWANA ARINA binti AKHIR. My matric number 23 DBT 20F2005. So, today I would like to do the simulation video about retractions, enzyme, digestions. In this simulation, you will be performing a retractions digest. You will use retractions enzymes to cut the DNA strands of two plasmids, producing DNA fragments because we know the size of our plasmid and where the enzyme will cut the DNA. We can predict the length of the fragments that will be produced. We can also confirm the length of these fragments or determine the length of fragments produced from an unknown plasmid constructs using gel electrophoresis. These DNA fragments can then be joined to make recombined plasmids. Next, this is the material needed for this experiment. First step is set the water bath temperature to 37 Celsius. Second step, select the P20 micropipette, then set the volume to 4 microliter and attach a tape to the micropipette. After that, move the retractions buffer tube to the empty tube rack and open it. Then, draw up 4 microliter of retractions buffer. Next, open the K+ tube. Then, dispense retractions buffer into K+ tube. Inject the tips into the track. Then, dispense 4 microliter of retractions buffer into the K- tube. Then, dispense 4 microliter of retractions buffer into the A+ tube. Next, dispense 4 microliter of retractions buffer into the A- tube. Next, dispense 4 microliter of retractions buffer into the A- tube. Next step is dispense 4 microliter of PKANR into the K+ tube. Then, dispense 4 microliter of PKANR into the K- tube. Next, dispense 4 microliter of PARA into the A+ tube. And dispense 4 microliter of PARA into the A- tube. and change the volume of the P20 micropipette to 2 microliter and dispense 2 microliter of filtration enzymes into the K plus tube. After that, dispense 2 microliter of filtration enzyme into the A plus tube and dispense 2 microliter of distilled water into the A minus tube. And dispense 2 microliter of distilled water into the K minus tube. Next step, open the microcentrifuge and place the solution tubes in the microcentrifuge. Then set the microcentrifuge to spin for 10 seconds. Remove the solution tubes. After that, open the water box and insert the floating solution tube route into the water box. And set the water box times for 1 hour. Lastly, open the freezer and place the samples in the freezer. Okay, this is the result of the experiment. Okay, that's all for me and thank you for watching.